Hi, namaste friends. So once again I'm here to make one more video on biomolecules. So in this video I'm going to talk about the enzymes. What are enzymes? So what are the properties of enzymes? What are the factors which affects on enzymes? Then classification of enzymes. So these are the points to be discussed under the enzymes. The first one is uh, enzymes. What actually enzymes? So you know that almost all enzymes are proteins. Many enzymes contain two essential components. One is protein part, another is non-protein part. Usually all enzymes are proteins. The protein consists of two components. Proteins ಅಲ್ಲಿ ಎರಡು ಪಾರ್ಟ್ ಇರುತ್ತೆ, ಒಂದು ಪ್ರೋಟೀನ್ ಪಾರ್ಟು ಅಂಡ್ ನಾನ್ ಪ್ರೋಟೀನ್ ಪಾರ್ಟ್ ಅಂತ. ಸೊ ಆಲ್ ದ ಎಂಜೈಮ್ಸ್ ಆರ್ proteins. The protein part is called apoenzymes, whereas the non-protein part is called coenzymes. You know the enzymes. So usually they are proteins. The that consists of two part or two components. One is protein part, another is non-protein part, other than protein part. The protein part is called apoenzymes, whereas the non-protein part is called coenzymes. In the examination, they may ask what is meant by apoenzymes. Otherwise, name the protein part of the enzymes. Name the protein part of the enzyme means apoenzymes. Non-protein part of the enzyme is coenzymes. So then, what actually enzymes? Enzymes are usually proteinaceous substances produced by living cells that catalyze biochemical reactions without themselves being changed in the process. So they are proteinaceous substances. Proteinaceous substances means the substances which consist of protein substances is known as proteinaceous substances produced by living cells. They are produced by living cells of the body and that catalyze biochemical reactions. That catalyze biochemical reactions means they helps in the rate of reactions. They may fasten or otherwise they may speed up the reactions. The rate of reaction has been increased with help of enzymes without changing in the process. It's called what? Enzymes are. Clear? Enzymes are the proteinaceous substances. They are produced by the living cells and that energize biochemical reactions or otherwise that catalyze biochemical reactions without themselves being changed in the process, and they are known as enzymes. Enzymes chemical substances ನಮ್ಮ ದೇಹದಲ್ಲಿ ಉತ್ಪತ್ತಿ ಆಗ್ತಾ ಇರುತ್ತೆ. ಸೊ ದಟ್ ಕನ್ಸಿಸ್ಟ್ ಆಫ್ ಪ್ರೋಟೀನ್. ಸೊ ದೇ ಆರ್ ನೋನ್ ಆಸ್ ಪ್ರೋಟೀನೇಷಿಯಸ್ ಸಬ್ಸ್ಟೆನ್ಸಸ್. ಅವೇನು ಮಾಡ್ತಾವೆ ಅಂದರೆ ರೇಟ್ ಆಫ್ ರಿಯಾಕ್ಷನ್ ಇನ್ಕ್ರೀಸ್ ಮಾಡ್ತಾ ಇರ್ತವೆ. ಬಯೋಕೆಟಲಿಸ್ಟ್ ಅಂತ ಕರೀತಾರೆ ಎಂಜೈಮ್ಸನ್ನು. ಈಗ ಫಾರ್ ಎಕ್ಸಾಂಪಲ್ ಕೆಮಿಸ್ಟ್ರಿಯಲ್ಲಿ ಲ್ಯಾಬೊರೇಟ್ರಿಯಲ್ಲಿ ಕೆಟಲೈಸ್ ಯೂಸ್ ಮಾಡ್ತಾರೆ. ಸೊ ರಿಯಾಕ್ಷನ್ ಫಾಸ್ಟ್ ಆಗಲಿ ಅನ್ನೋದಕ್ಕೆ. ಸೊ ಹಾಗಾಗಿ ಎಂಜೈಮ್ಸ್ ಆರ್ ಲೈಕ್ ಎ catalyst. So they catalyze the metabolic activities. They may increase the rate of reactions which takes place in the body of organisms. So they are protein substances. They catalyze the biochemical reactions. So what are the properties of enzymes? So they are protein nature. Number one is protein nature. Protein nature means all are proteins. So they are having proteinaceous substances. Colloidal nature. Colloidal nature means sticky nature anta. Generally hydrophilic colloids anta. So they are sticky in nature. Then third one is unchanged form anta. Unchanged form means without changing themselves they involved in chemical reactions anta. ಅಂದರೆ ಅವು ಫಿಸಿಕಲ್ ಸ್ಟೇಟಸ್ ಆ್ಯಂಡ್ ಕೆಮಿಕಲ್ ಸ್ಟೇಟಸ್ ಯಾವುದೇ ಚೇಂಜ್ ಆಗೋದಿಲ್ಲ, ಆದರೆ ದ ಮೇ ಹೆಲ್ಪ್ ಇನ್ ರೇಟ್ ಆಫ್ ರಿಯಾಕ್ಷನ್ಸ್ ಅಂತ. ರೇಟ್ ಆಫ್ ರಿಯಾಕ್ಷನಲ್ಲಿ ಹೆಲ್ಪ್ ಮಾಡ್ತಾ ಇರ್ತವೆ, ಆದರೆ ಅದರಲ್ಲಿ ಯಾವುದೇ ಕೆಮಿಕಲ್ ಸ್ಟೇಟಸ್ ಆಗಲಿ ಅದರ್ವೈಸ್ ಫಿಸಿಕಲ್ ಸ್ಟೇಟಸ್ ಚೇಂಜ್ ಆಗ್ದೇ. ದಟ್ ಈಸ್ ವಾಟ್ ಅನ್ಚೇಂಜ್ ಫಾರ್ಮ್. ದೋ ಇವನ್ ಇನ್ವಾಲ್ವ್ಡ್ ಇನ್ ಕೆಮಿಕಲ್ ರಿಯಾಕ್ಷನ್ಸ್ ದೇ ಆರ್ ನಾಟ್ ಚೇಂಜ್ಡ್ ಅಂತ. ದೇ ಆರ್ ನಾಟ್ ಚೇಂಜ್ಡ್. ಅವು ಕೆಮಿಕಲ್ ರಿಯಾಕ್ಷನಲ್ಲಿ ಪಾರ್ಟಿಸಿಪೇಟ್ ಆಗ್ತವೆ, ಆದರೆ ಅದರ ಯಾವುದೇ ಕೆಮಿಕಲ್ ಪ್ರಾಪರ್ಟೀಸ್ ಫಿಸಿಕಲ್ ಪ್ರಾಪರ್ಟೀಸ್ ಚೇಂಜ್ ಆಗೋದೇ ಇಲ್ಲ. They may involved in a chemical reactions without changing themselves. So that is called what? Unchanged form. Clear? The first one is protein nature. They are proteinaceous substances. Second one is colloidal nature, sticky nature. Third one is unchanged form anta. So they are not changed even involved in chemical reactions. Thermosensitivity. Thermosensitivity means so they are very sensitive towards temperature. If temperature rises more, so enzyme will be inactive, or otherwise if temperature falls less, again enzymes become inactive. So they may active only during optimum temperature. Optimum temperature means average temperature ಅಂತ. ಟೆಂಪ್ರೇಚರ್ ತುಂಬ ಜಾಸ್ತಿ ಇದ್ದರೆ ಎನ್ಸೈಮ್ಸ್ ವರ್ಕ್ ಆಗೋದಿಲ್ಲ, ಅದರ ತುಂಬ ಕಡಿಮೆ ಇದ್ದರೆ ಎನ್ಸೈಮ್ ವರ್ಕ್ ಆಗೋದಿಲ್ಲ. ಸೊ ಟೆಂಪ್ರೇಚರ್ ಅವರೇಜ್ ಇರಬೇಕು. when temperature average, then only enzymes will be active. So that is what thermosensitivity. So thermosensitivity means they are very sensitive towards temperature. Then pH sensitivity. So very sensitive towards pH concentration. Even pH level also. pH level also should be on average. If 2 pH, otherwise less pH, they will be inactive. Team activity. They usually enzymes work together. So these are the certain properties of the enzymes. Clear? So they are protein substances, colloidal in nature, unchanged form, thermosensitivity, pH sensitivity, and team activity. Team activity means they may exhibit togetherness. So they may work in all together. Factors affecting enzyme activity. See, there are plenty of factors which affects on enzyme activity. What are those factors? The examination they may ask, mention the factors which affects on enzymes ಅಂತ. ಎಂಜೈಮ್ ಮೇಲೆ ಪರಿಣಾಮ ಬೀರ್ತಕ್ಕಂಥ ಅಂಶಗಳು ಅಂತ. ಯಾವ ಅಂಶಗಳು ಎಂಜೈಮ್ ಮೇಲೆ ಪರಿಣಾಮವನ್ನು ಬೀರ್ತವೆ? There are a number of factors which affects on enzymes in order to increases the rate of reactions. Enzymes show highest activity at certain pH and temperature known as optimum pH and temperature. See, enzymes show highest activity at certain pH and temperature known as optimum pH and optimum temperature. Optimum pH means ಯಾವ ಪಿ ಎಚ್ ಲೆವೆಲಲ್ಲಿ ಎನ್ಸೈಮ್ಸ್ ವರ್ಕ್ ಆಗುತ್ತೋ ಅಂಥ ಪಿ ಎಚ್ಚನ್ನು ಆಪ್ಟಿಮಮ್ ಪಿ ಎಚ್ ಅಂತ ಕರೀತಾರೆ. ಆಪ್ಟಿಮಮ್ ಟೆಂಪ್ರೇಚರ್ ಅಂದರೆ ಯಾವ ಟೆಂಪ್ರೇಚರಲ್ಲಿ enzymes will be work, that temperature is known as the optimum temperature. Clear? So that is what optimum pH and optimum temperature. Above or below to this, the enzyme activity decreases. Above or below this means for example the pH more than optimum, enzymes will be inactive. Otherwise below optimum pH, again enzyme activity will be inactive. Otherwise temperature more than optimum temperature, enzymes cannot work. Otherwise below optimum temperature, enzymes cannot work. So enzymes will be work at optimum temperature only. Enzymes will be work at optimum pH only. If more temperature, otherwise less temperature. If more pH level, otherwise less pH level, enzymes cannot work. ಗೊತ್ತಾಯ್ತಲ್ವಾ? ಆಪ್ಟಿಮಮ್ ಟೆಂಪ್ರೇಚರಲ್ಲಿ ಮತ್ತು ಆಪ್ಟಿಮಮ್ ಪಿ ಎಚ್ ಅಲ್ಲಿ ಮಾತ್ರ enzymes will be work. Otherwise enzymes cannot work. Lower temperature decreases the enzyme activity. Lower temperature decreases the enzyme activity. Lower temperature means less temperature below optimum temperature. That may decreases the enzyme activity. Enzyme activity na ಕಡಿಮೆಗೊಳಿಸುತ್ತೆ. And it remains temporarily inactive and higher. So enzyme will be inactive when temperature falls down. Temperature degenerates the enzyme and higher temperature denatures the enzymes. See, lower temperature decreases the enzyme activity and it remains temporarily inactive. and higher temperature denatures the enzymes. ಸೊ ಜಾಸ್ತಿ ಟೆಂಪ್ರೇಚರು ಸಹಿತ ಎನ್ಸೈಮ್ನ ಡಿನೇಚರ್ ಮಾಡ್ತಾ ಇರುತ್ತೆ. ಇವನ್ ಲೋಯರ್ ಟೆಂಪ್ರೇಚರು ಸಹಿತ ಎನ್ಸೈಮ್ ಆಕ್ಟಿವಿಟಿನ ಡಿಕ್ರೀಸ್ ಮಾಡ್ತಾ ಇರುತ್ತೆ. ಸೊ ಆಪ್ಟಿಮಮ್ ಟೆಂಪರೇಚರ್ ಈಸ್ ವೆರಿ ಮಚ್ ಎಸೆನ್ಷಿಯಲ್ ಫಾರ್ enzyme activity. Clear? So optimum pH and optimum temperature are the major factors which affects on enzyme activity. Generally, enzymes act in a narrow range of temperature. At 0 degree, the enzyme activity is minimized. Usually, enzymes will be active at narrow range of temperature. Narrow range of temperature means optimum temperature. ತುಂಬ ಜಾಸ್ತಿ ಟೆಂಪ್ರೇಚರ್ ಅಲ್ಲ, ತುಂಬ ಕಡಿಮೆ ಟೆಂಪ್ರೇಚರ್ ಅಲ್ಲ, ಅವರೇಜ್ ಟೆಂಪ್ರೇಚರನ್ನು ನಾವು ನ್ಯಾರೋ ರೇಂಜ್ ಆಫ್ ಟೆಂಪ್ರೇಚರ್ ಅಂತ ಕರಿತೀವಿ. ಅಂಥ ನ್ಯಾರೋ ರೇಂಜ್ ಆಫ್ ಟೆಂಪ್ರೇಚರ್ ಮಾತ್ರ ಎನ್ಸೈಮ್ಸ್ ವರ್ಕ್ ಆಗ್ತಾ ಇರುತ್ತೆ. ಎನ್ಸೈಮ್ಸ್ ವಿಲ್ ಬಿ ವೆರಿ ಆಕ್ಟಿವ್ ಅಟ್ ನ್ಯಾರೋ ರೇಂಜ್ ಆಫ್ ಟೆಂಪ್ರೇಚರ್. ಅಟ್ 0 degree C, the enzyme activity is minimized. As the temperature increases, the enzyme activity is also increases. ತುಂಬ ಟೆಂಪ್ರೇಚರ್ ಜಾಸ್ತಿ ಆದರೆ ಎಂಜೈಮ್ ಆಕ್ಟಿವಿಟೀಸ್ ಆಲ್ಸೋ ಇನ್ಕ್ರೀಸಸ್. ಅಟ್ 30 to 40 degrees C is active, but more than 45 degrees C, enzyme activity is retarded. See, 30 to 40 degrees C is considered as optimum temperature. If more than 40-45 degrees, then enzyme activity will be decreased. If below 30 degrees C, again enzyme activity will be reduced. So 30 to 40 degree is considered as optimum temperature where enzyme will be work. Even pH also. Every enzyme shows maximum activity at a neutral pH. Example, pepsin and trypsin 8.5 pH. So when pH falls at a neutral, then only enzymes will be work. Otherwise enzymes cannot be work. Clear? So this is what the factors which affects on enzyme activity. Inhibitors, chemicals that bind to the enzymes and decreases or inhibits activity are called enzyme inhibitors. Enzyme inhibitor means the substances which inhibits enzyme activity, which suppress the enzyme activity are called inhibitors. Inhibitors ಅಂದರೆ ಯಾವ ಸಬ್ಸ್ಟೆನ್ಸಸ್ಸು ಎಂಜೈಮ್ ಆಕ್ಟಿವಿಟಿನ ಸಪ್ರೆಸ್ ಮಾಡುತ್ತೋ, ಇನ್ಹಿಬಿಟ್ ಮಾಡುತ್ತೋ ಅಂಥವನ್ನು ಇನ್ಹಿಬಿಟರ್ ಅಂತ ಕರೀತಾರೆ. ಕ್ಲಿಯರ್? The chemicals that bind to the enzyme and decreases or inhibits its activity are called enzyme inhibitor. Otherwise, in simple terms, the chemical substances which inhibits means which decreases, which reduces the enzyme activity, and they are known as inhibitors. And clear? And the process is known as inhibition. The process where enzyme activity will be inhibited by the certain chemical substances are called inhibition. So such chemical substances are called inhibitors. Clear? So this is about what inhibitors. Then classification and nomenclature. How the enzymes will be classified. How enzymes are nomenclatured. According to International Union of Biochemistry, enzymes are classified into 6 major classes. According to International Union of Biochemistry, the enzymes are classified into major classes based on their functions. Oxidoreductases, hydrolases, transferases, liases, isomerases, and ligases. So this is how the enzymes can be classified. According to International Union of Biochemistry, enzymes are classified into 6 major classes based on their functions. Number 1 is oxidoreductases. So what is the function of oxidoreductases? They catalyze the addition or removal of hydrogen or oxygen between two substrates. And substrates means the substances on which enzyme will be activate. That substances is known as a substrates ಅಂತ. ಯಾವ ಸಬ್ಸ್ಟೆನ್ಸ್ ಮೇಲೆ ಎಂಜೈಮ್ಸ್ ಆಕ್ಟಿವಿಟಿ ಎಕ್ಸಿಬಿಟ್ ಮಾಡುತ್ತೋ ಅಂಥ ಸಬ್ಸ್ಟೆನ್ಸನ್ನು ಸಬ್ಸ್ಟ್ರೇಟ್ ಅಂತ ಕರೀತಾರೆ. Here they catalyze, they means oxidative. It is enzymes which catalyze the addition or removal of hydrogen or otherwise oxygen between two substrates. ಎರಡು ಸಬ್ಸ್ಟ್ರೇಟ್ ಮಧ್ಯೆ either removal of hydrogen, otherwise addition of hydrogen, or otherwise removal of oxygen, or otherwise addition of oxygen. So these are it takes place between two substrates. So which is done by oxidoreductases. Then second one is a hydrolysis. So addition of water molecules to a bond of the substrate. So this is the function of hydrolysis. Hydrolysis function is the addition of water molecules to a bond of the substrate. Then transferases. Transferases means a transfer of group from a donor to an acceptor. So this is what the function of the transferases. And here transferases means, so the enzymes which transfer of group from a donor to acceptor, donor ಇಂದ acceptor ಗೆ ಟ್ರಾನ್ಸ್ಫರ್ ಮಾಡಿದ್ರೆ, so that is the function of transferases. Then lyases. Lyases is also one of the enzymes. These enzymes remove groups from the substrates without using water leaving double bonds. See, these enzymes remove groups from the substrate. Substrate ಇಂದ ಗ್ರೂಪ್ ಅನ್ನು ರಿಮೂವ್ ಮಾಡ್ತಕ್ಕಂಥ ಎಂಜೈಮ್ಸು ಲಯೇಸಸ್ ಅಂತ. ಲಯೇಸಸ್ ಆರ್ ದ ಎಂಜೈಮ್ಸ್ which remove the group from the substrates without using water leaving double bonds. Isomerases. These enzymes are involved in interconversion of optical, positional and geometric isomers. The enzymes which are involved in interconversion of optical, positional and geometric isomers are the responsible of isomerases. And last one is ligases. Ligases are the enzymes that link two different compounds by making bonds such as carbon oxygen, otherwise carbon sulfur, otherwise carbon nitrogen or phosphorus oxygen bonds. These enzymes link two different compounds by making bonds. So that is what ligases anta. So likewise based on the function of the enzymes can be classified anta. So this classification which is proposed by International Union of Biochemistry. So that classify the enzymes into 6 major classes based on their function. So that is oxidoreductases, hydrolyses, transferases, lyases, isomerases and ligases. So these are the classifications of enzymes. Clear? Then cofactors. What actually cofactors are? See here, so in the examination they may ask anta. So mention the classification of enzymes and otherwise mention the names of the mention the types of the enzymes and so these classifications are very important. Then cofactors. What actually cofactors? Enzymes may have either one or more than one polypeptide chains. Some have only protein part, some others have both protein and non-protein parts. So usually we discussed the enzymes are proteinaceous substances. There are two components. One component is protein part, another component refers to non-protein part. Protein part and the protein iruttade anta. Non-protein part andre maybe other than protein part, either carbohydrates, either fats or otherwise vitamins or otherwise any other molecules except protein substances, protein part as well as non-protein part. Enzymes that have only protein part are called simple enzymes. The enzymes which consist of only protein part and they are known as simple enzymes. That have both protein and non-protein part are called conjugate enzymes or otherwise hollow enzymes. And the enzymes which consist of only protein part and they are known as simple enzymes. Certain enzymes which are having both protein part as well as non-protein part and they are known as conjugate enzymes or otherwise hollow enzymes. And here in the examination they may ask what are simple enzymes or otherwise what are hollow enzymes or conjugate enzymes. And enzymes which are having protein part and they are known as simple enzymes, and enzymes which are having both protein and non-protein part are called conjugate enzymes. So that we can consider enzymes consist of protein part also, even non-protein part also, but majority of enzymes are proteinaceous in nature. Hollow enzymes. Whole enzyme means enzymes which are having both protein as well as non-protein. For example, apoenzymes and cofactors are collectively called hollow enzymes. And the protein part is called apoenzymes and non-protein part is called cofactors. And so you know that simple enzymes. Simple enzymes means the enzymes which are having only protein part are called simple enzymes. Conjugate enzymes are otherwise hollow enzymes. It means enzymes which are having both protein as well as non-protein part are called hollow enzymes. And so that hollow enzymes is nothing but protein part as well as a non-protein part. So apoenzymes and cofactors, so both are together called as a hollow enzymes. Apoenzyme means protein part, whereas cofactors means non-protein part. And in the examination they may ask name the protein part of the enzymes or otherwise non-protein part of the enzymes. And the protein part of the enzymes are apoenzymes, non-protein part of the enzymes are cofactors. Clear? So in the first slides itself we told that there are two part. And so here there are any enzymes contain two essential components, protein part and non-protein part. So protein part is called apoenzymes and non-protein part called coenzymes. Clear? Likewise here also. And the cofactors means hollow enzymes. Hollow enzyme means which are having both protein as well as non-protein part. Protein part is called apoenzymes, whereas non-protein part is called cofactors. For cofactors needed for catalytic activity of enzymes. Cofactor means both protein as well as non-protein part. Cofactor means non-protein part. All enzymes means both protein as well as non-protein part. Only protein part apoenzymes or otherwise simple enzymes. So both protein and non-protein conjugate enzymes, otherwise hollow enzymes. Cofactors is needed for catalytic activity of enzymes. There are three types of cofactor are prosthetic group, coenzymes and metal ions. Cofactor means non-protein part. So there are three types of cofactor. One is prosthetic group, coenzymes and metal ions. So prosthetic group, coenzymes and metal ions. So these are all the types of cofactors. Clear? So what are cofactors? So name the protein part of the enzymes, non-protein part of the enzymes. Likewise they may ask. So cofactors means non-protein part, whereas protein part apoenzymes. So certain enzymes consist of only protein part and they are known as simple enzymes. Certain enzymes consist of both protein as well as non-protein part and they are known as hollow enzymes or otherwise conjugate enzymes and clear. Non-protein part is called cofactors. So there are three types of cofactors: prosthetic group, coenzymes, and metal ions and clear. So this is about what the cofactors and then classification and nomenclatures and so there are six major classes based on functions: oxidoreductases, hydrolases, transferases, liases, isomerases, and ligases. So these are the types of enzymes. Then inhibitor, the substances which are inhibit the enzyme activity are called inhibitors. So factors affecting enzymes. There are two important factors which affects an enzyme activity. One is pH, another one is temperature. Optimum pH and optimum temperature. Optimum pH means average pH, whereas optimum temperature means average temperature. Not too much and not too less. So then only enzymes will be active. Clear? Then properties of enzymes: protein nature, colloidal nature, unchanged form, thermosensitivity, pH sensitivity, and team activity. So these are the properties. Clear? Enzymes, meaning of enzymes. So this is about what are the enzymes. And in the examination they may ask what are enzymes and for 1 mark definitions. So enzymes are the proteinous substances that energize chemical reactions or otherwise enzymes are proteinous substances that catalyze chemical reactions without being changed themselves and ಅವು ತಮ್ಮಷ್ಟು ತಾವು ಚೇಂಜ್ ಆಗದೇ ಕೆಮಿಕಲ್ ರಿಯಾಕ್ಷನ್ನ ಫಾಸ್ಟ್ ಮಾಡ್ತಾ ಇರ್ತವೆ, ಅದರ ಸ್ಪೀಡ್ ಅಪ್ ಮಾಡ್ತವೆ. ದೇ ಸ್ಪೀಡ್ ಅಪ್ ದಿ ಕೆಮಿಕಲ್ ರಿಯಾಕ್ಷನ್ಸ್. So such substances are known as enzymes and clear. The properties of enzymes also very important. They are proteinaceous in nature, colloidal in nature, unchanged form, pH sensitivity, thermosensitivity, and team work. They work together. So these are the properties of enzymes. And then classification of enzymes. Classification of enzymes. So classification, there are 6 types based on their function. That is what classification of enzymes. Then so usually enzymes can be identified with suffix of as ಅಂತ. See suffix of as, liases and ACS, isomerases ASE, ligases ASE. So the word, the suffix of ASE, then that will be considered as enzymes ಅಂತ. ಯಾವ ವರ್ಡು ASE ಇಂದ ಕೊನೆಗೊಳ್ಳುತ್ತೋ ಅದು ಎಂಜೈಮ್ಸ್ ಅಂತ. ಈಗ ಲೈಸಸ್, ಐಸೋಮೆರೇಸಸ್, ಲಿಗೇಸಸ್, hydrolases, transferases, oxidoreductases. So these are the enzymes. Clear? Then cofactors. The enzymes which consist of protein part are called simple protein or otherwise apoenzymes, whereas enzymes which consist of both protein as well as non-protein called, and they are known as conjugate protein, conjugate enzymes or otherwise hollow enzymes. So all enzyme is equal to apoenzymes plus cofactors. Protein part of the enzymes is called apoenzymes, whereas non-protein part of the enzyme is called cofactors. There are 3 types of cofactors. One is prosthetic group, second one is coenzymes and metal ions. And prosthetic group, coenzymes and metal ions. So these are the types of cofactors. And clear? So this is about what the enzymes. And clear? So friends, so in this video, so we have finished biomolecules. With this I have finished biomolecules. The biomolecules chapter is closed. See, in these biomolecules, the important points are the meaning of biomolecules, types of biomolecules, then micromolecules, macromolecules. What are biomicromolecules? Given examples. What are biomacromolecules? Give examples. Then structure of micromolecules: amino acids, glycerol, fatty acids, sugar, pentose sugar, hexose sugar. These structure are very very important. Whereas in macromolecules, protein structure, polysaccharides, so are the very very important. Then enzymes. In this video, so I have finished enzymes, properties of enzymes, then factors affecting enzymes. Last one is cofactors. Clear? So we need to concentrate on this. So with this I'm going to finish this video. So by next week I'm going to start new chapters in the next video. So until that, thank you friends, thank you very much, good luck.